The use of stem cells in science gives us insight into subjects such as cellular control, cancer, and even aging. The Jasper Lab is focused on regulatory mechanisms that control stress tolerance, metabolism, and aging with the help of fruit flies. In particular, Dr. Jasper has been recognized for making seminal discoveries about the effects of aging on stem cell behavior and the role of stress in regulating stem cell function. Current projects in his lab focus on the control of tissue regeneration, metabolic homeostasis, and cell death by insulin and stress signaling pathways. Here we learn how science moves from a bench in a lab at a school into an industrial clinical setting.
Don't know much biology.
Hello and welcome to Radiobio. I'm your host, Morgan Quayle.
And I'm your co-host, Christy Donham. We are joined here today by Dr. Heinrich Jasper, a researcher at the Buck Institute.
How are you today?
Good. Thank you for inviting me.
So, do you mind telling us a little bit about yourself?
Yes, I was trained in Germany originally. I did my undergraduate studies at Tübingen University, which is in southern Germany, and my Ph.D. then in Heidelberg, which is still in southern Germany. And then started the lab pretty much right out of my Ph.D. In the biology department of the University of Rochester, where I stayed on as assistant professor and then became tenured associate professor in 2010, before then being recruited to the Buck Institute for Research on Aging in Novato in California in 2012. There I spent 5 years continuing our work on aging and stem cells in fruit flies and now in mice as well. And just a few months ago, I decided to do another move, this time to Genentech, which is, of course, a company that uses a lot of basic research in order to develop new therapies for a wide range of very complex diseases, including inflammatory diseases.
I know you mentioned stem cells. How would you define a stem cell and how they're used in your research?
So we are primarily interested in what we would call somatic stem cells or tissue stem cells. These are generally cells that are in a somewhat undifferentiated state, and they have the ability to both self-renew, which means they can generate their own type of cell again, but they can also go and generate cells that move on to differentiate and regenerate the more functional cells of a tissue.
Where do these stem cells reside? Are they all over, or is it something that's super localized?
It depends on the tissue. Every tissue has a little bit of a different configuration. You know, in the blood system, the hematopoietic system, you find stem cells in the bone marrow. So it depends on the tissue, but often the tissues that are exposed to a lot of damage, for example, barrier epithelia like the intestine or the airway, they do have a very active and or very productive stem cell compartment, right? And this is a stem cell compartment that does change a lot with age and with disease, and something that we're trying to explore in detail.
What led you to study stem cells? There's such a broad area, and so
we got to a point in our research which was initially focused on the genetics of aging. And these genetic factors are often relatively broad regulators of metabolism and stress tolerance. But in order to understand their role, we had to go and understand their mechanism of action in a particular tissue, in a particular cell type. We figured that the best way of really exploring the consequences of disrupting these type of genes would be to explore them in the context of a process that is really critical for maintenance of a youthful function, or of what we would call homeostasis, in a complex organism. And to us, that process is clearly regeneration, because if you think about it, in order to really extend lifespan, or in order to really maintain a youthful state, the only thing you really need to do is to allow the tissue to really exert the same regenerative potential that it has had when you were born.
So actually, I'm kind of interested in where you get these stem cells, because often when we hear about it in the news or something, we're talking about human stem cells. So where do you get your stem cells of interest for your work?
So we're looking, as I mentioned, we're looking at somatic stem cells and tissue stem cells. And what this means is we're actually going into the animal and look in their tissues for these stem cells, which means the experiments we're doing are ultimately targeted genetic interventions, which means that we are perturbing gene function or protein function within the stem cells in the living organism.
A few minutes ago, you just mentioned how we can use stem cells to study aging. And after listening to your talk, I was really interested in what you mean by that. So how do your studies actually relate to aging?
The question we're trying to address is why we are losing the ability to properly repair our tissues. Basically, you have to think about it this way, right? The stem cells have intrinsically the capacity to generate and regenerate every single functional cell of a particular tissue. Why is it then that in an older animal or in an older human, this ability declines? Because if it wouldn't decline, the tissue would basically be maintained forever. I'm not saying we're going to live forever, but we would be able to maintain tissue health without much problems, because any damaged cell could be replaced immediately, right? But that's clearly not the case. And so in the context of aging, we do need to understand why this capacity, this regenerative capacity, declines of disease.
I actually heard you talk about how stem cells have the ability to migrate. We often think of cells as stationary items in our body. What does it mean when you say that they're actually migratory?
Well, what I mean by this is that when we look at them, they move. And so we don't understand the reason for their migratory behavior yet, but they do move in relation to other cells in the same tissue. They change position. They move away from each other. They join with other cells. There's clearly it's clearly a very dynamic system, but it's very early to really make any conclusions about the significance of this and whether the movement that they have has anything to do with the regenerative potential, with the repair process itself, or whether it's an observation that is independent of their normal function. So that's something we're still exploring.
I just want to know so much about that. Like, how did they do this? Like, why did they? Because normally you think of stem cells as like the basal layer, right? The foundation.
Right.
And you'd think it wouldn't move. Like a foundation on a house, if it were to move, the whole house would crumble. So then how do they, in my head, I'm just thinking of this as, and it kind of blows my mind that it moves and it's so versatile. Like, how do they do it and why? But
these are all good questions, and we're very excited about asking these as well. And so the good thing is we have a system in the fly where we can explore exactly these questions, right? We can ask about the significance of this movement for the regenerative process. We can ask about mechanism, about how do they move and why do they move, and how do they stop moving. We can ask about whether a migratory cell is better in regenerating an enterocyte, which is a functional cell of the intestinal epithelium, or whether it can also regenerate an enteroendocrine cell, which is a hormone-secreting cell. All of these questions are open, and we don't know, and we'll explore this in the next few years, hopefully. Human disease. That's a good question. I mean, the reason we're using flies and mice is that, first of all, the flies are a great system technically in the lab, which allows us to very rapidly perform very complex experiments, actually, where, as I mentioned earlier, we're disrupting gene function in a particular cell type at a particular time, and then we're asking what happens to the tissue. Flies, of course, are somewhat removed from vertebrates, and so evolutionarily, and so we are using the mouse as a way to ask whether the mechanisms that we have identified in the fly can also be observed in or are at work in in vertebrate systems as well. And of course, humans are vertebrates, right? So by comparing the fly with the mouse, we're already jumping in the evolutionary scale quite broadly, and we're really asking about conserved common mechanisms between very distant species. And so fundamentally, if we want to develop an intervention to target a human disease, we will have to do the experiment where we take what we have learned in the mouse, generate an intervention strategy, test it again in the mouse, and then potentially go into humans. So one thing that we're trying to get into now and we are working on is to also, as an intermediate step, maybe have so-called organoid studies where we are taking human stem cells out of their tissues, and there are protocols now that have been developed in many different labs that allow us to develop small organs that replicate some of the complexity of the human organ in a dish.
We wanted to know a little more detail about what an organoid is, and found that an organoid is a three-dimensional, miniaturized, and simplified version of an organ. It is produced in a lab environment and has the potential to show realistic microstructures and function. Scientists can use these organoids to test potential human treatments. Dr. Jasper goes a little further into details on this process.
Right, so we have small intestinal organoids, for example, then where we have a structure that looks like a small gut with stem cells in the particular areas where they should be in the gut itself, with a lumen that contains some of the mucus and the proteins that we usually find in the gut lumen of humans. We don't have bacteria in there yet. Maybe that's something we will do soon. And so in these kind of small organs, these organoids, we can then ask questions about, for example, drug treatments that we think should affect stem cells or should rejuvenate stem cells, and ask whether these drug treatments would really have the function that we expect them to have in a human context. And that would be the next step before then going into the patient, right?
How do you get those human stem cells to put into these organoids?
Well, we isolate them, for example, from human patients that have where you have to take biopsies, usually during clinical studies or in treatments for disease. It is possible to obtain biopsies from the gastrointestinal tract, for example, or during colonoscopies, for example, you get biopsies, or during bone marrow transplants, you of course get the bone marrow that contains a lot of hematopoietic stem cells, although there's really no organoids for hematopoietic stem cells. But from bronchial biopsies, you can grow airway organoids. From intestinal biopsies, you can grow intestinal organoids. From muscle biopsies, you can grow myofibers in the dish. So there's a number of ways we can get this tissue, and we can then isolate the stem cells from this tissue and then grow it in culture. And they actually spontaneously develop into organoids.
So what's next for you, and how do you come up with your next question?
So we will continue to work to try and understand how aging affects stem cell function. But going to Genentech now, my lab is going to keep working on the fundamental questions, and these questions about how stem cells are regulated and how aging impacts that regulation from different aspects, continuing to focus primarily on intestine and airways, and how the immune system is interacting with these stem cell populations, but then also trying to apply that knowledge to the development of therapies for diseases that are often associated with aging, but are also ultimately a condition of chronic inflammation. And these include diseases like asthma and COPD, and chronic obstructive pulmonary disorder, or inflammatory bowel disease, or idiopathic pulmonary fibrosis. All of these are disease indications that Genentech is very interested in, and that we believe we can contribute to understanding with the studies we're doing on age-related dysfunction in these stem cell systems.
So, in your work, to get to this therapeutic stage, do you think you'll get there by the end of your career? Or, like, how far away are you from that?
Well, generally, it's interesting, actually, because many people have asked me, and I've asked myself, of why would Genentech bring in a group that works in flies, or even a group that, in general, works on very basic questions, right? And that's where Genentech is really visionary in a sense, because they believe that understanding the basic biology of disease processes and of homeostasis is, in the long run, going to allow them to develop innovative therapies that nobody else has developed. But they're fully aware that this is a process that takes 10, 15, 20 years. So at the end of my career, I still have a few years to go, but I'm assuming that I will hopefully have contributed to new ideas and new strategies, and potentially even to new targets and at least new trials for intervention. Whether we will ultimately get to a productive drug, who knows? What I do know is that at Genentech, I would be in the best place possible to at least attempt that, because they have a track record of developing really effective therapies that are based on a rational approach. And this is an exciting step.
When I'm thinking about your work, 50, 100 years from now, I'm imagining some sort of dystopian future where everyone can have access to synthetic organs.
We're already doing that.
That is happening, yeah.
Yeah.
Not in my lab, but it's but people are definitely thinking about this and are interested in developing new organs. And it's obvious, right? I mean, we are in dire need, for example, for organs for transplantation. I mean, lung transplants are essential for certain diseases. And we just don't have enough. And to develop a lung, or develop an artificial kidney, or to develop a heart that is truly functional, we could not do better than to replicate what we already have. And to do so, we would have to use these basic biological approaches to really generate a real organ. And once we can do this, yes, we should be able to replace the heart easily, and to replace maybe the liver, maybe the lung, maybe the kidney. I'm not sure about the brain yet, but we'll see.
So if we can regenerate tissues and now organs, why can't we just live forever?
Well, I don't think one needs to necessarily propose that, but what is clear, I believe, is that by understanding this and by finding ways of regenerating tissues, we should easily be able to maintain a healthy life for a century or so, right? So basically age until you're 95, and then maybe something gives, but we're still healthy, right? And this is really ultimately the goal, right? Is to basically try and reduce this period of morbidity where we are slowly declining. And which can be very painful both for the individual as well as for the families. We would really want to try and find a way to reduce that period and therefore live a long and healthy life.
Another question, this is more of a fun question. I know when I worked in fly labs before, every day smelled like I was in a candy shop. What does it smell like when you walk in your lab? What does it feel like when you walk in every day?
I always feel like it smells more like beer. At least it makes me feel happy. But it can be an acquired taste, right? And the thing is, it's fly food, and it consists mostly of yeast and sugar, and so it does taste like a bakery or a brewery, right?
Mm-hmm.
That's really what it is. Some people like it a lot. Some people hate it. So.
And I know I love it.
Yeah, so that's good.
And then kind of one of our favorite questions, what are some misconceptions about your research?
The larger misconception, I think, is more in our aging work, where the question often becomes, well, do you subscribe to the idea that we want to live 1,000 years, right, or live forever? And I don't think, and I think that's the wrong question to ask, actually. I think because the question about immortality is often fraught with a lot of misunderstanding of what biology really is. It is unlikely that that will happen for the simple reason that our bodies are very complex systems that have to maintain an open thermodynamic state in order to maintain that complexity. So it is a very difficult proposition. Would it ever be impossible to do that? I don't know, because if we look at our germ cells, they do live forever in one way or another. They do rejuvenate themselves and generate the next generation, right? So biological systems do have that intrinsic capacity in some way or another, but the real question is whether our somatic bodies, these very complex, differentiated systems, can ever be redesigned in a way that allow them to not decline in function. And the question is, would we ever want that? Because it might require us to change the makeup of our bodies in a way that is impossible to fathom and would ultimately lead to changes that nobody really wants for themselves. In the aging field, the consensus has emerged that what we want and what the NIH is also supporting is research that will lead to this compression of morbidity and an expansion of what people call health span, but that efforts to live forever are not really on the radar at this point, and are usually, when people discuss these, are usually mixed up with philosophical misconceptions of what life and what biology really is.
Radiobio is supported by the Quantitative and Systems Biology Graduate Group and the Graduate Division at the University of California, Merced. For more information, you can visit our website at radiobio.net or follow us on Facebook, Twitter, or Instagram.